good lineup of speakers. If we can have a first slide, first presentation. Okay, so this is the Host-Pathogen Interaction session. I have to thank all the speakers. So I'll be the first speaker, in fact. I'm the chair and the first speaker. I have to thank Ricardo for the invitation. It's really a great pleasure to be here. Ricardo, although I'm probably the only one that's not on the network of interactions that Elder showed yesterday. I have no papers with Ricardo, but definitely Ricardo had a great influence over my career, as many of us have testified over these days. So it's really a pleasure to be here. Thank you, Ricardo. Thanks for all the help you've given me along the years. Because of the subject of the symposium of climate change and infectious disease and host pathogen interactions and immune responses, I decided to talk about something we've been doing recently in the lab that has more to do with this really monitoring, trying to monitor these complex interactions in the field and then bring them into the lab so we can try to understand some of the mechanisms behind of these interactions. So I'll tell you a little bit about our recent studies, trying to understand the impact of Wolbachia and then the symbiont bacteria on the virome of Aedes aegypti mosquitoes. And so basically, okay, why do we care about mosquitoes? We've heard about them over the past few days. Mosquitoes, they transmit a lot of viruses that are very important for human health. So here I have listed the top 4: dengue, yellow fever, Zika, and chikungunya. They're all transmitted by the same types of mosquitoes. Basically, they are Aedes mosquitoes, mostly Aedes aegypti for us, but in some regions, Aedes albopictus. And these mosquitoes, they are really widespread globally, which means these diseases can be transmitted almost across the whole globe. And here is just a map that shows the public health burden of dengue. So although this map is from 2013, it's actually very updated. If we think about the incidence, basically the epicenter of arboviral diseases are South America and Southeast Asia. And we heard yesterday, I was glad to see data from Tulio showing arboviral circulation in Africa, because we really lack data. So these viruses are spread across the world. Dengue alone is estimated to cause about 400 million infections every year. So it's a huge number of infections. In Brazil, in 2023, we had about 5 million cases, confirmed cases, which we estimate is about 1/10 of the real number of cases. So with 5 million, it means 50 million cases. A quarter of the population was infected in a single year. So this is an incredible burden for the health system. And of course, climate change will impact transmission of these viruses. Basically, on the best case scenario, we will have an increase of about 2.4 billion people that will be in areas of transmission. With the worst case scenario, which seems more likely these days, we're going to have another 5 billion people exposed to transmission of mosquito-borne viruses. So I work in Europe, and now we have local transmission of dengue and chikungunya in Europe. So in Southern Europe, there's already local transmission. Of course, they have a couple hundred cases, which for them is a big impact. But if we compare to 5 million cases in Brazil, it's nothing. But this will increase with climate change. So what can we do? Here is a cycle of arboviruses. So the mosquito acquires the virus from a viremic person, and then the virus has to actively grow in the mosquito, reach the salivary gland, where it can be then transmitted to a naive individual upon the next blood feeding of the mosquito. Which means the virus has a complex cycle. Maybe not as complex as some of the parasites we heard over the past few days, but it is very complex for a virus because it has multiple hosts. And here I'm just showing human-to-human transmission, but we could include animal reservoirs. So this can be very complicated. And for most of the arboviruses, vaccines and specific treatments are not broadly available, or they are not efficient enough, or they are not safe enough. We've heard about the yellow fever vaccine, for example. And in fact, the fact that the virus can grow in the mosquito also poses a threat that even if you immunize the whole population, can the virus persist only in the mosquitoes? So these are questions we still don't know. So from the perspective of my lab, we think that vector-based strategies are also required. So we need to target not only the human patient, but also the vector. And here is a very interesting success story targeting the vector. The only vector intervention strategy shown to have an impact on arbovirus transmission in the wild. If you think insecticides had an impact or population reduction, there is no scientific data showing that, okay? There is a lot of trends, but no scientific data. For Wolbachia, a randomized controlled trial was carried out in Indonesia, and the introgression of Wolbachia, the bacteria and the symbiont, into mosquito populations led to a reduction of 77% in dengue incidence. So if we couple that with vaccination and other strategies, maybe we can really eliminate dengue, right? And how does it work in the case of Wolbachia? It's what we call population modification strategy. So Wolbachia is a bacteria that infects the germline of the mosquito. And when a mosquito carrying Wolbachia crosses with a mosquito without Wolbachia, all the progeny carries Wolbachia. So basically, if you release a few mosquitoes here in blue, for example, in wild populations, they will cross with the wild population. In a few generations, basically, they will all carry Wolbachia. And what happens is mosquitoes that are naturally susceptible to the arbovirus they transmit, once they carry Wolbachia, they become resistant. So you are shifting your population of mosquitoes for virus susceptible to virus resistant, which means they won't transmit viruses. That's the simple idea behind it. What's the problem here? As there are questions about the mechanism of action, sustainability, and potential ecological impacts of Wolbachia introgression into wild Aedes aegypti populations. So although Wolbachia is present in over 60% of all invertebrate species, naturally, it's not present in Aedes aegypti. So it's an artificial introduction. And every time we play with nature, we don't know what will come back to us, right? So we really need to understand that better. And so the goal of my lab is really to try to understand how Aedes aegypti mosquitoes acquire, host, and transmit arboviruses. And here what's interesting is that we've focused across the years on many factors, but recently we came across the fact that the viruses that are in the mosquito, so they're natural viruses or persistent viruses, if you will, they are usually specific to the mosquito, so they are not transmitted to us. They have a very strong interaction with the host. This is what we call the virome. So we are very used to hearing about how the microbiota is very important for the host, but we don't hear the same amount of information about the virome. So what's the virome? Virome is basically the collection of viruses circulating in mosquitoes, right? This includes the arboviruses, but also the insect-specific viruses, so viruses that never leave the mosquito. But they don't leave the mosquito, but they are definitely influencing the physiology of the mosquito, and they can interact with arboviruses. So we've published recently, so this is one of the very interesting observations we have, where we identified two ISVs, Humaitatubiacanga virus and Fazicaroni-like virus, that when present in the mosquito population, they increase dengue transmission by 50-fold. So that's a very, very important epidemiological impact. So the presence of two natural viruses of the mosquito can impact transmission of dengue to humans. So clearly we need to understand more about how they affect the host in this case. And as I said, Wolbachia does the opposite. So when you introduce Wolbachia, you decrease the amount of infected mosquitoes. When you have HTV and PCLV, you increase the amount of infected mosquitoes with arboviruses. So we have different trends in viruses that naturally circulate in mosquitoes and Wolbachia that's being introgressed artificially. So how these things will interact in the wild, we have no clue. And so really, I think the introgression of Wolbachia into mosquitoes will have an impact on the virome, and this can impact the host in ways that we're not expecting. So remember, all the strong mechanistic data with Wolbachia came from experiments in the lab where these viruses do not exist, right? We've shown in our work that basically, if you look at lab colonies, they lack viruses. They don't carry natural viruses. They are all found in the wild. So here, what I'll show you was our attempt to characterize the impact of Wolbachia introgression on the virome of Aedes aegypti and the consequences to arbovirus transmission. So to do that, we chose one site of the releases of mosquitoes containing Wolbachia by the World Mosquito Program. So the group of Luciano Moreira is carrying this out throughout Brazil. And Niterói was one of the first sites where they released mosquitoes starting in 2017. And they divided basically the city of Niterói, which is just across Rio de Janeiro. And they released in different zones, and they kept a control zone where mosquitoes with Wolbachia were not released. Bear in mind, this is not a randomized controlled trial, so it doesn't have the same impact, but they observed some potential reductions in arbovirus transmission. But to us, that was not what was important. We really wanted to know what was happening with the dynamics of virus circulation in the wild. So here is the data that they generated about the releases. So in Zone 1, the releases started in January 17, Zone 2, July 17, Zone 3, December 17, Zone 4, October 19. And here's the frequency of Wolbachia. And you can see basically the frequencies are related to when they started releasing, because this was tested in 20. So you can see Zone 4 started in October 19th. In 20, it was very low. So we obtained samples from basically these samples, and from these areas, because they had a biobank of mosquitoes that they collected before a release and during the releases. So our question is, as Wolbachia was introgressed, what happened to virus circulation? And so we had samples from 18, 19, 20, and 21, and we performed basically metagenomic analysis of these mosquitoes to identify which viruses were present. We also analyzed Aedes albopictus that were living in the same areas and collected in the same traps, but I'm not going to talk about this data today. And so what we performed is a metagenomic analysis that we developed in the lab that's based on small RNAs that really is designed to identify viruses, and especially novel, highly divergent viruses that might be present in mosquitoes. And so when we did that to our samples, we identified 6 viruses circulating in Aedes aegypti in Niterói, and 1 in albopictus that I won't talk about it. What you can see here is that there is a big diversity of viruses, but they're all RNA viruses. So we had 2 positive-stranded RNA viruses, 3 negative-stranded RNA viruses, and 1 double-stranded RNA virus. So this is really what we tend to find in mosquitoes. So there's nothing very different there. In terms of how these viruses were detected, one important thing, we didn't detect any arboviruses. But this is not a surprise, again, because arboviruses are found in very low frequencies in wild mosquitoes. Usually 1% of mosquitoes carry arbovirus, so it's really hard to pick them on these types of studies. But we found all these 6 viruses. And you can see even these 6 viruses had very different prevalence and load in the mosquitoes. So here is the viral load for HTV, PCLV, Quaranja virus, Guadalupe mosquito virus, a Virga virus, and the Almendra virus. And you can see some of them were detected very consistently across all samples, across time and zones, while some had some drops and some were rare. Like detected, these 2 were rare, were detected in just a few samples. For the ones that were detected very consistently, like HTV and PCLV that I told you before affect transmission of dengue, we had enough samples to do some analysis. And so what we observed was an inverse correlation between the load of Wolbachia and the load of HTV and PCLV. So there was an inverse correlation between the ISVs and Wolbachia, which was very interesting. So, of course, this involves a few samples that we perform metagenomic analysis. But we wanted to know if we increase the numbers, if we really have high numbers of samples, can we go beyond these simple correlations? And so we went back to the samples, and we analyzed 663 individual mosquitoes from April, May 2021. So we had one point in time that we could analyze a large number of samples. And so with these samples, we can detect, of course, Wolbachia. And you can see here what I showed you before from 2020, from Luciano's data, that there is an increased frequency of Wolbachia in the zones 1, 2, and 3, where the releases start in 17, a lower frequency in 19, and an even lower in the control zone where it was not released by the time. So we have now zones with different amounts of Wolbachia where we can now do some statistics on the presence of the viruses. And so on this, I'll skip quickly because I'll have a summary quickly afterwards. But basically, we identified the effects of Wolbachia on PCLV, HTV, OVV, not GMV, but also Quarandia and Almendra, and here is just the summary we think is more important. So if we look at the populations of mosquitoes without Wolbachia and with Wolbachia, what you can see is that there is a significant difference on the prevalence of HTV, PCLV, and OVV. So all three viruses were significantly decreased by the introgression of Wolbachia. So Wolbachia affected them negatively the same way it affects arboviruses. So that was going on along the trend, let's say, of antiviral activity of Wolbachia. But then for GMV, we observed no effect. So the virus had the similar circulation independent of Wolbachia. And for the Quarandia virus and the Almendra virus, they are rare, as you can see, in terms of the prevalence compared to the others. They had the reverse effect. They had an increased circulation when Wolbachia was introgressed. So that was really kind of unexpected. But this was basically reduced to an impact on the prevalence. If we look at the viral load of these viruses in the mosquitoes with and without Wolbachia, when the virus was present, the viral load was basically independent of Wolbachia. But Wolbachia overall was changing the prevalence. So it's really affecting the transmission, but not the replication in the infected individual. And so here, what we did odds ratio analysis to see the association, the strength of the association between WML, Wolbachia, introgression, and the insect-specific virus. And as you can see from it's very similar to the previous data, for OVV, HTV, and PCLV, there is a very significant reduction in the incidence or prevalence of these viruses in mosquitoes when Wolbachia is present. And for the Almendra virus and the Quarandia virus, there is a very significant increase in the chances of finding these viruses in the presence of Wolbachia. And if we turn this around and we use a logistic regression here to now ask, can we correlate the probability of Wolbachia infection with the detection of the ISVs? Yes. So here we see that when we detect the Quarandia virus and the Almendra virus, there is a significant increase in the chances that Wolbachia is present. And when we detect PCLV, HTV, and OVV, there is a significant chances of not detecting Wolbachia. So there's really a strong association between these viruses and Wolbachia. So here, our data really shows a very strong impact of Wolbachia on the natural virome of Aedes aegypti. Clearly, the structure of the virome is changing completely after introgression of Wolbachia. But one thing that we cannot parse out from this data is interactions between the viruses. So, of course, if Wolbachia affects one virus and this virus affects the other virus, Wolbachia will affect the other virus indirectly. So our data cannot discriminate the possibilities. So there's all these associations between Wolbachia and ISVs, but there are associations between the ISVs themselves. So if a mosquito is infected with PCLV, there is a 685% chance of that mosquito also being infected with HTV. So there is a strong association between these viruses. On the other hand, if the mosquito is infected with HTV, there is a 87% chance, a lower chance of that mosquito carrying the new Quarandia virus we identified. So there is very complex interactions in the wild that's very difficult to understand, and that's why we bring these things to the lab. So our results suggest this impact, but it doesn't tell us how it happens and how it affects the mosquito. So that's why we bring these things into the lab to try to understand. So here, although we just started, we have very interesting preliminary results that I'll share with you on that I think are very important. So the way we do is we bring the wild mosquitoes into the lab, and we rear them as individual females. So they will lay eggs from individual females. They'll be tested for the ISVs, and from this, we can separate colonies that carry 1, 2, 3 ISVs, as many as you want, with and without Wolbachia. So we can separate really the colonies the way we want, all derived from the same wild-type mosquitoes. And the wild-type mosquitoes came from the Wolbachia factory here in Belo Horizonte. So with that, afterwards, we can then treat the mosquitoes with tetracycline and generate the same line with and without Wolbachia. So all the ISVs with and without Wolbachia. And for the sake of time today, I'll focus on the most interesting virus that we found, which is the Quarandia virus. Remember, it had a positive association, strong positive association with the introgression with Wolbachia. But this virus is really rare. So here we analyzed about 1,400 public libraries, RNA sequencing libraries, to determine which libraries derived from mosquitoes this virus was present. And basically it was present in 27 libraries out of 1,400, so 2%. 2% of all the mosquitoes sequenced in the world carry this virus, so it's a very rare virus. For example, PCLV is detected in over 30% of them. But the metadata of these samples told us a lot about this virus. First, it was Aedes aegypti specific. So all the libraries derive from Aedes aegypti. And this, we often find that with these insect-specific viruses, that they are species-specific. So they don't cross species. So that was interesting. It was not present in any of the Aedes aegypti cell lines, so only in vivo, only in the adult mosquito. And it was detected, out of the 27 samples we detected, 24 came from Australia. And why is that important? Because the mosquito strain where Wolbachia was first introduced was from Australia. So basically, we think this virus is a carryover from the original introgression of Wolbachia that was spread throughout the world. So this is something very important that we're going after now, because basically, with the releases of mosquitoes with Wolbachia, we're also releasing this new virus that we know very little of. So this is something we are looking at. And so here is a line we developed that carries just the Quaranja virus. So it's free of all the other ISVs here by RT-qPCR detection, but it carries high levels, so over 90% of the individuals with high levels of the Quaranja virus. And see here, when we eliminate Wolbachia compared to the original line that had Wolbachia, that has no impact on the Quaranja virus. So the association we see is not an association with Wolbachia. So Wolbachia does not seem to favor this virus. It's just a matter of piggybacking along the way. What we think is that Wolbachia definitely affects the other ISVs, and this ISV is competing with the other ones. So when Wolbachia eliminates the other ISVs, it allows this ISV to proliferate. And why is this important? For many reasons. Probably it will affect the mosquito. And here is just an example. So it's preliminary results, but an experiment we did in the lab exposing these mosquitoes to blood feeding on mice. And you can see here mosquitoes without the Quaranja virus, they blood feed more efficiently than mosquitoes that carry this Quaranja virus. So is that important? Oh, that looks good, right? They mean they're going to feed less. But remember, if a mosquito does not complete blood feeding efficiently, it will feed multiple times. So in terms of transmission, these mosquitoes might be more efficient at transmitting. So we don't know. Basically, we need to study more. So basically, in summary, Wolbachia really affects the virome of Aedes aegypti. And this led us to identify this novel Quaranja virus. So Quaranja virus is an automixo virus. So it's, let's say, the flu of mosquitoes. And it is associated with Wolbachia introgression, but the virus is naturally very rare, which means we really need to understand that we'll be modifying the populations with this virus now. So our data really highlight the need that we have to evaluate more closely the impact of these interventions. And with that, I have to stop and thank the people that did the work. So all the bioinformatics was done by João Paulo here in the audience with the help of Victor on the Wolbachia part. And the experiments were done by Matthias and Izaki, also here in the audience, with help from other people in the lab. And our collaborators, we got great help from mathematicians in the UK, Luigi and Mario. So very suggestive names. So Mario and Luigi are our collaborators. And here in Brazil, of course, the World Mosquito Program, this was all done in collaboration with them. All the biobank came from them. And with Álvaro here at Fiocruz and our financial support, including Ricardo here that, reluctantly financed the mosquito studies. So, and with that, I'll stop here and take questions. Thank you.
Okay, so I apologize if I missed this, but I was under the impression that Wolbachia only colonized one sex of the mosquito. And so is it true that does that impact the virome of a male mosquito versus the female mosquito?
So it also colonizes males. Of course, the transmission is efficient through the ovaries, but it also colonizes males. So it not only colonizes the germline, but other tissues as well. And the impact is the same on males and female mosquitoes for the virome, exactly the same. That was very surprising to us, is that when we separate the data by sex, it's identical.
Thanks for a great talk, really fascinating. So this is almost a personal thing for me because my wife was involved in the Gates program,
Okay.
the original Wolbachia, and I have a picture of her releasing mosquitoes in Queensland. So, but my question is, and you may have touched on this, and again, I might have missed it too, is there any evidence that there's pressure on the arboviruses themselves to adapt to the Wolbachia resistance?
Yes.
And is there enough Wolbachia out there in the mosquito population that you might expect to have mutations in the pathogens?
Yeah. So that is the biggest concern at the moment with Wolbachia, is the sustainability, exactly because of that, Alan. So in experiments in the lab and in the field, what you see is that eventually you find strains that grow well in the mosquitoes with Wolbachia. So we don't understand the mechanism, especially because we don't understand how Wolbachia affects the virus. So it's really, we're still unclear, but there are strains that grow very well. And the estimates is that the older Wolbachia-released mosquitoes can contain the virus for about 10 years. And after 10 years, they will be adapted. That's why we need something else to come at the same time. So that's why the vaccines, they were betting on the vaccines. And we need now, clearly, another alternative to Wolbachia. Otherwise,
those viruses have been sequenced?
they've been sequenced, but so all the strains have been sequenced and they don't find common mutations. So it's not very clear why they escape.
João, that was fantastic, thank you. So it's more related to the mechanism by which the Wolbachia impacts the virome. And I think for the dengue, it's still unknown, right?
Unknown.
So,
well, depends who you ask. But
Yeah.
for me, it's unknown, yeah.
But I mean, if you think about the other viruses, especially the ones that like replicates more in the presence of Wolbachia, so you think the mechanism is something related to specific signaling in the or virus interaction in the microbiome of the mosquito is just a matter of competition with other viruses. So as Wolbachia inhibits some virus, or this other virus can now replicate because there is more niche for replication there. So what's your
Yeah, I think for the Quarandia virus, it's probably a competition, really just a traditional competition with the other ISVs. Right. But Wolbachia, for me, Wolbachia also competes with the virus. So I think the best explanation for how Wolbachia inhibits viruses is they compete for the same resources in the same host. And Wolbachia is an intracellular bacteria, so it requires a lot of contribution from the host, just like viruses. And possibly, you know, it impacts the metabolic patterns of the mosquito, and this affects the virus. So thank you very much. We can move on to our next speaker, which is Dario Zamboni from USP in Ribeirão Preto. Thank you, Dario.
So first of all, I would like to congratulate Ricardo again. This is a beautiful history of like supervising students and setting up this amazing collaboration group. So, and then I would like to thank the organizers and Ricardo for the invitation as well to speak a little bit about our work with the coronavirus and the infection. So I will start with this slide, which, I mean, COVID needs no introduction. This is just like a slide that shows the waves of like virus in Brazil up to October 2022. So this is the ancestral Wuhan variant, this is the Gamma variant, and this is the Omicron. This is cases, this is deaths, and thanks to the vaccination in green, I mean the deaths is highly reduced, although the number of cases are super high in the Omicron variant. And then this is a little bit of what we saw in Brazil in terms of the distribution of these infections. And then most of the data I'm going to talk today is from patients that were infected in the early stages of the infection with the Wuhan variant of SARS-CoV-2. So just to make the point, I mean, Brazil was severely hit by the pandemic. I mean, we had about 2.7 of world population, and then we had more than 10% of the deaths due to SARS-CoV-2 infection. So then I will start with the early evidence indicating that the inflammasomes are associated with the pathogenesis of SARS-CoV-2. The first study published in May 20 with 21 patients from Wuhan, they associated the severity of the disease with the cytokine storm and macrophage activation syndrome, suggesting that there was a highly inflammatory situation in this fatal cases of SARS-CoV-2 or COVID-19 infection. And then later on, in June 2020, there was this other paper, little more patients, 107 patients, also from Wuhan. And then they associated LDH, which is lactate dehydrogenase, which is an intracellular enzyme, which during inflammatory process, they are released when there are lytic forms of cell death. And then in the serum of these patients, there was super high LDH release, indicating a lytic form of cell death. So this leads the whole community to understand the role of the inflammasomes in the pathogenesis of SARS-CoV-2. And then I just gonna very brief introduction about the inflammasomes because of the time. But then inflammasomes are these platforms that are in the intracellular receptors that stays in an inactive form in non-infected situations. As soon as we have an infection going on, there is a conformational changes of this receptor, and there are many sensors of the inflammasomes, NLRP3, M2, NLRC4, NAPES, and so on. But they all oligomerize it somehow, and then they recruit this small adapter protein called ASC, which binds to the amino-terminal domain of the receptor, and then there is formation of filaments via perin-perin interaction of the ASC, and the card domains of the ASC stays from the external part of the filaments, and card domains they stick to other card domains, and then there is a formation of aggregate, which we call puncta or speck, which is actually the active inflammasome. As soon as there is infections, then or inflammatory process going on, they aggregate and they formate this beautiful puncta. And then in this structure, there is still card domains, which are ready to cleave and recruit and cleave pro-caspase-1, which is the inflammatory caspase. Then it's cleaved, and now we have active caspase-1. And caspase-1 cleaves pro-IL-1 beta, which is also produces as a proenzyme, or pro-IL-18, which induces the active formation of the active forms of IL-1, IL-18. And then, of course, caspase-1 also cleaves this gazermin D, which is this pore-forming protein that punches the pore in the cells, and through the pore, IL-1 beta, IL-18 can go out, and the cells swell, the water goes in, and then they completely collapse, and there is inflammatory form of cell death. So LDH, which is in the intracellular, goes outside, and all the inflammatory cytokines as well. So this is a very important response that we have against infections. For instance, for bacterial infection, they are very protective. This is a picture. This is 4 pictures from a movie in that we infected macrophages with Legionella pneumophila, which was one bacteria we like to study in the lab. And Legionella is, this is a macrophage. You can see green because we stained it NLRC4, which is one of the inflammasomes that senses flagellin. And then Legionella, this macrophage is phagocyting Legionella. You can see them in red here. And then as soon as the video reaches 1 hour of the infection, there is a formation of the inflammasome. You can see right here. And right in the sites of the inflammasome formation, there is gazermin D pore formation in these membranes of the cells. And then propydium iodide, this is in the media right here, they goes inside and then stains the nuclei. You can see the membrane blabbing. And then this is 5 minutes different, this picture from this picture. So the cells completely collapse, they swell, and then they release inflammatory process. And then bacteria can no longer replicate in this dead cell, and then actually in the middle of a lot of membranes of the dead cell. So it's a very efficient mechanism to control bacterial infections, and thanks to these platforms, we are resistant to Legionella. Most of us are resistant to Legionella. We breathe this bacteria, we don't get sick because of this very important innate immune responses, which restricts bacterial replication. But in case of virus, we had studied a Mayaro virus before in chikungunya, and they kind of the inflammasome doesn't work as expected. They kind of tend to worsen the scenarios in patients, and this was exactly what happens in SARS-CoV-2. I'm going to lead to this data now. As I mentioned before, all these patients, the first study we worked with 124 patients that seek for health assistance in the hospital of the School of Medicine in Ribeirão Preto. And then we work with so because they reach hospitalization, most of them were moderate and severe cases. We had 34 deaths in these patients. So, and all in the beginning of the pandemic. We got like samples from the serum of these patients in the day of the hospital internalization, and then we can see, comparing the healthy controls with the COVID SARS-CoV-2 patients, infected patients, you can see that there's a high active caspase-1. This is a active caspase-1, which means inflammasome is highly active in these patients. IL-18 is super high in these patients as well. IL-6 as a proof of principle that IL-6 was known before to be highly expressed in the patients of SARS-CoV-2, of COVID-19. And then we can, of course, correlate IL-18 with caspase-1, which makes sense. Caspase-1 is cleaving pro-IL-18, so one is high, the other one is high as well. Caspase-1 with IL-6, IL-18 with IL-6. So all these inflammatory mediators were super high and nicely correlated. So then we managed to get samples from the lungs of this patient. We can see highly expression of NLRP3, infected cells in the lungs of these patients. This is autopsy of the lungs. And you can see the actual inflammasome speck formation, or the puncta formation, indicating the active inflammasome in the lungs. And I'm going to get back to these autopsy studies in the end of this talk. So, and then we managed to correlate the magnitude of the inflammasome activation at the day of the hospital admission of these patients, and then we can see that the patients that required mechanical ventilations, like the worst cases, and then they had higher levels of IL-18 as compared to the patients that did not require mechanical ventilation. If you separate the patients between severe COVID and mild severe, there's a higher caspase-1 P20 in the patients with severe COVID, and then the same for survivors versus non-survivors. And then we can see there's higher inflammasome activation in the non-survivor, the patient that did not survive due to SARS-CoV-2 infection. And then we did also longitudinal studies, and then we found that the patients that died due to COVID-19, they reached the hospital with super high levels of inflammasome activation in the red line right here. Even though they reduced it during the hospitalization, they didn't make it, and then associating the inflammasome with the outcome of SARS-CoV-2. This was a paper published at the beginning of the pandemic. It was accepted in the end of 2020. This Tamara Rodrigues was the first author that carries on this study. She was a second-year PhD student. She did amazing work with a lot of in collaboration with clinicians and many different labs in the School of Medicine in Ribeirão Preto. So then we move on to, the question is the inflammasome just correlating with the worst case scenarios? Is the cause of the severity of SARS-CoV-2 and leading to death and to worst case scenario? This is different possibilities, and then the right way to answer this question is using mouse deficient for NLRP3 and see how the infection goes without the gene that actually activates the NLRP3 inflammasome. So we managed to generate this NLRP3 knockouts back-crossed to humanized ACE2, so we can infect this mouse with the real SARS-CoV-2. And then we can see here that the parenchyma area of the mouse infected is super inflamed, so this means like inflammation in response to viral infection. And when we did the same experiment with NLRP3 deficient mouse, you see that it's much better than as compared to the wild-type mouse. And all the clinical parameters like body weight and clinical score, they are much better in the absence of NLRP3, and they are more protected to the death due to SARS-CoV-2 infection, indicating that the inflammasome is also promoting the pathogenesis that leads to the worst case clinical outcomes in COVID-19. So then question is like the basics, how the NLRP3 inflammasome is activated in response to the virus. SARS-CoV-2 has many different ORFs and non-structural protein that some of them are porins. We tested a lot of the expression of these several genes from the SARS-CoV-2 in macrophage. We could never find a reliable gene that induces inflammasome activation. But then NLRP3 inflammasome can be activated by a canonical pathway, which happens when there is a pore formation in the membrane of the cell, but also it can be activated by a non-canonical pathway that requires mouse caspase 11 or human caspase 4. So this is a non-canonical pathway for NLRP3 activation. And then we found that the SARS-CoV-2 was really activating the non-canonical NLRP3 pathway, which requires mouse caspase 11 or human caspase 4 is the same gene. So then this is experiment infecting macrophages with SARS-CoV-2. We pull down active caspase 11. Caspase 11 is not canonical cleaved like caspase 1, so we have to pull down these active caspases with biotin VED, and then we Western blot against caspase 11. So we can see this is active caspase 11 in response to SARS-CoV-2. This is just input. We can also show the requirement of caspase 11 for this NLRP3 activation, because when we infected macrophages deficient for caspase 11 in the red arrow right here, you can see there is no inflammasome activation. This is the control for this, the wild type IL-1 beta activation, but not in the IL-18 deficient. And then also scoring NLRP3 punta, which is the inflammasome activation. It's highly reduced in the absence of caspase 11, suggesting that caspase 11 is indeed a pathway by which the NLRP3 inflammasome is activated in SARS-CoV-2. This is just the same experiments with mouse deficient for caspase 11, backcrossed to human ACE2. So you can see in the red arrows right here, in the absence of caspase 11, the mouse gets better situation when infected. This is the mock and infected controls, highly inflamed lungs by the loss of airways in the parenchyma of the lung of this mouse, and this is much better in the absence of caspase 11. And also in the survival curves, and caspase 11 survives better than the wild type of caspase 1 plus or caspase 11 plus minus littermate controls. So, and then just like we got a help from Elder to analyze data from patients infected with SARS-CoV-2 and a super high expressed caspase 11 in the COVID-19 patients, and correlate with inflammasome other genes like caspase 1, correlates with caspase 4, which is mouse caspase 11, and IL-1 beta with caspase 4, indicating that this is actually happening in patients as well. So that's idea that the inflammasome is really activated via the non-canonical pathway, and also other groups also show that the caspase 11 plays a role in activation of the non-canonical pathway in response to SARS-CoV-2. And now I'm going to switch to this data that we have with 47 fatal cases of SARS-CoV-2. There's a very fantastic pathology in the School of Medicine, which is Alexandre Fabro. He managed to got samples from the lungs of the patients in the moment of the death or right after death, and collecting small samples that we could analyze and see. And again, this is all these fatal cases were obtained in the beginning of the pandemic, which is important because there is no variants, no vaccine, so we could really see how the disease occurred in the ancestral SARS-CoV-2 variant. So then first thing Kayla did was look to the inflammasome activation in the lungs, and we saw a huge distribution for the patients, some of them very low NLRP3 activation, some of them high. And for many cells, we look at the NLRP3 or the punta of ASC and NLRP3 are super high in the patients as compared to the healthy or infected controls. So we can see here that this is really the inflammasome. We stained ASC right here, we stained NLRP3, and they nicely correlate as expected, indicating that this is really the NLRP3 inflammasome, which is active in the lungs of patients that deceased due to COVID-19. So then Kayla did a lot of expression analysis in these patients, and we could clusterize these patients in two groups very clearly. One of them, the cluster 2, is highly inflamed. The cluster 1 is the opposite, not very inflamed. Viral load is super high in cluster 1 as compared to the cluster 2. And I mean, this is NLRP3 punta is super high in the inflamed as expected, but not in the cluster 1. You can see also illness time. This patient takes more time. This is the patient that the cure of the viral replication, there is a very low virus in this cluster 2 patients, and they stay in the UCI for many months or many days, and eventually they die longer times as compared to the cluster 1. The respiratory situation of this patient is much worse. The cluster 2 as measured by PF radio and AA gradient, so you see much higher difficulties in breathing of these patients from the cluster 2, which is according to the idea that they are more inflamed. So this is the same during the hospitalization time of these patients. So this summarizes all these studies with the autopsy of these patients. Expression of inflammatory genes super high in the cluster 2, inflammasome activation super high, opposed to the cluster 1, time to death higher in the cluster 2, respiratory status worst in the cluster 2, pulmonary viral load super high in cluster 1, and then we found more fibrosis in the cluster 2 patients, and then the reason of the question we took a lot of time to respond is why the patients from the cluster 1 are dying. They are not so inflamed. What happens with this patient? And then we found a lot of disseminated intravascular coagulation in the lungs of the patients from the cluster 1. So it's pretty much SARS-CoV-2, the ancestral variants without vaccination. There is the two outcomes in the fatal cases, right? So everybody die here because it's all autopsy. So then one group of patients, super inflamed situation. The other one's more intravascular coagulation problem regardless of the inflammatory process. So not only inflammasome promotes the death of the patients, but also intravascular coagulation in some other patients. So you can see here 3 samples, 3 from different patients from cluster 2. You can see erythrocytes in red, super high in the patients from the cluster 1, in agreement with the disseminated intravascular coagulation, and much higher fibrosis. You can see collagen in green right here in these patients, indicating higher fibrosis and higher inflammatory process in these patients. So then one aspect that I think it's very interesting to study now is having these samples from the autopsy is look to the inflammasome in other cells. So most of the inflammasome community works and study macrophages. This is clear that in SARS-CoV-2, macrophages are the main cells that activates inflammasome and promotes the inflammatory cytokine storm and everything we know about inflammatory process in SARS-CoV-2. But we see inflammasome in many other cells as well. So that's, this is macrophages, of course. This is ASC puncta. I hope you can appreciate this. Maybe this light is too strong, but there is a puncta right here. There's a puncta right here in NLRP3 and ASC. Endothelial cells, the same. We can see a lot of active inflammasome in endothelial cells as well in SARS-CoV-2. And we also see inflammasomes in type 1 and type 2 pneumocytes in the epithelial cells. And we compared these patients, the autopsy of this SARS-CoV-2 patient with patients that died to influenza. And then we found that the inflammasome activation also occurs in influenza lethal cases. But then the inflammasome activation in influenza is much more present in type 1 and type 2 pneumocytes as opposed to SARS-CoV-2, which is much more present in macrophages and endothelial cells. So kind of the inflammasome are there, they activate, but depending on the virus, intercellular like preferences, style, and replication niches for the cells, they have like different magnitudes of inflammasome activation. And then one thing I'm going to just finish with some unpublished data we are carrying on right now. So it's that Kayla, she compared ASC and NLRP3 puncta in many different cell types. And then she came up to me and said, well, there's these ciliated cells in the trachea. They are very present in the trachea. That doesn't match. I mean, there is much higher ASC, lower NLRP3 puncta, but a lot of ASC puncta in the, so it's kind of like a different inflammasome. It's not NLRP3 because in all cell types we use it, NLRP3 matches the ASC. So they're together, which means they are the NLRP3 inflammasome. But in these ciliated cells, there's a lot of NLRP3 puncta, ASC puncta, which is not NLRP3. And then I said, well, we should be M2 because M2 is another inflammasome highly active in these viral infections. It senses double-stranded DNA, different than NLRP3. And then bingo, Kayla bought the antibody, and then we stained ASC puncta, and then they match. And then ciliated cells, they have a lot of ASC M2 colocalization, indicating activation of the inflammasome. And these cells are very present in the trachea. The epithelial cells right here is much enriched. There is also some in the bronchi and bronchiola, but most of the cells, ciliated cells, they are in the trachea, which I think it's interesting because there is cells ready to recognize virus. there. And now we establish a model, a mouse model that we backcross at ACE2 with AIM2, and then we can see here, I hope you can appreciate here, the green is tubulin, so you can see the ciliated cells right here in this trachea. AIM2 is there, and then you can see 3 different regions of this wild-type mouse infected with SARS-CoV-2. We can see AIM2 puncta, and now we are studying this and ongoing with the infection study, as I showed you before, with parenchyma area, survival curves, and so on, to evaluate the role of this inflammasome, the AIM2 inflammasome, in the outcome of these infections as well. And I can score very nicely that in the absence of AIM2, there is no puncta, which is suggesting that this indeed the NLRP3 AIM2 inflammasome in this trachea of this mouse. And then just to summarize a little bit of our contribution of the study of the inflammasomes during SARS-CoV-2, we first published that the NLRP3 worsened the several cases of SARS-CoV-2 infection. This process goes via caspase-4, the non-canonical pathway for NLRP3 activation. The fatal cases can be divided in 2 main groups. The one highly inflamed, one super inflammatory, high inflammasome activation. The other one is more problems with vascular coagulation, which also is lethal and lead to death of the patients. As inflammasome is so important to the several cases of COVID-19, we screened it for a library of compounds to search for a drug that targets inflammasome. I'll not show you this data, but we found like some drugs that really target the activation of the inflammasome and then can be used as a treatment of SARS-CoV-2. We published this in 2022. And then ongoing studies using this virus, both influenza and SARS-CoV-2, as a model to understand the inflammasome activation in non-macrophage cells, and they are really important as well to contribute to the inflammatory process in response to the virus. And with that, I would like to thank the really people that deserve all the acknowledgement in this work. There's a fantastic group in School of Medicine in Ribeirão Preto and many funding agents that support the work made in our lab. And just to point out that the amazing work made in my lab, the viruses were led by women. So there's like Tamara, she's now a professor in Federal University of Santa Catarina. Kayla, she's now doing a postdoc at Yale. Leticia is still in the lab. Adrienne and Amandinha, did an amazing job in SARS-CoV-2 investigation in the lab. So with that, I'd like to thank you guys for the attention.
Thank you very much, Dario. We have time for a couple questions. So maybe while Dario's walking, I can start. So it's interesting that you just showed an example of a DNA sensor activated by an RNA virus. So that's common. That's been shown for sting, important for some RNA virus. So what's the trigger for M2 activation coming from a double RNA virus?
Yeah. So we did not go to this direction, but my response to this is that my feeling is that there's mitochondrial damage and mitochondrial DNA is activating in response to NLRP4. So that's in M2 inflammasome. So I think it will be M2, and this may have sting activation as well.
So if I got you right, the NLRP3 inflammasome is activated. That has a major effect. AIM2 inflammasome is activated. That has a major effect.
I don't know if there's a major effect AIM2.
Well, you had no specs.
No specs. Yeah, I don't know if the survival is going to be different, the pathology, the lung situation, inflammation. The contribution of I would say, Doug, contribution of the AIM2 inflammasome should be much lower than NLRP3. This is my expectations. And there is beautiful work by Judy Lieberman showing that AIM2 is also activated in the lungs of the patients and so on. So I think they are both activated. The effect of this activation for the outcome of the disease, I would say NLRP3 is the inflammasome that is worse in the scenario.
All right. Well, you answered my question. So thanks.
Sorry.
Absolutely wonderful, Dario. Can you elaborate a little bit more at the very end? Doug and I were just talking about this. The field of inflammasome and activators is a big field right now. And as you know, Luke O'Neill and lots have companies that are actually doing this. Have you actually started to think about a clinical trial? And my second question is, how much of this is variant specific? If you look at the later variants, are they giving you more inflammasome activation, less inflammasome? And then finally, the effective vaccination, is it really protecting us from this kind of disease?
Yeah, that's great questions, Alan, so thank you. So I'll start with the third question, is that the inflammasome is, the infection goes on, and then most of the research in the lab we work with macrophage pathogens like Legionella and Leishmania, and then we always look to the macrophages. And the funny story is that we started looking to the virus because of the early evidence indicating that inflammasome was there, and we had all the patients in the right place at the right time, so then we should do that. And now that the COVID, SARS-CoV-2 wave passed it through, and then we still have this virus and this material, and then we start looking to other cells. And influenza is very clear because there's very, very few inflammasomes in the macrophage. They're all in pneumocytes. So that would be nice models for us to understand this. So I may keep the line of investigation for the influenza. But for the COVID, all these variants and strains and the vaccination, then I thought it was too complicated for us to keep in this field, highly competitive. Maybe now that the United States cut all the funding to SARS-CoV-2, and Brazil, SARS-CoV-2 is not only BCL3 anymore, it's BCL2. So maybe it's time to go back to SARS-CoV-2. But it's a very interesting point then.
Thank you very much, Dario. We have to move on. So we'll have a little change to the program. We'll have Rafaela Ferreira from the Department of Biochemistry and Immunology from here, UFMG, presenting now. And then we'll get back to the normal program after the coffee break. So Rafaela, thank you.
Thank you. Good morning, everyone. It's a great pleasure to be here. I've been hearing a lot about very exciting research, mainly on vaccines and now vectors, pathogenesis. And what I'm going to tell you about is mostly about drug discovery. So how are we approaching some of the molecular targets that are important for several neglected diseases? And in our lab, we have been working mostly with proteases. Proteases are very important targets. These proteins are about 2% of our genome, and they are important for humans in several processes. They are also important for pathogens. And we have several successful stories of the development of protease inhibitors as drugs. A few examples here are the angiotensin-converting enzyme, DPP-4 for diabetes, HIV proteases, hepatitis C virus, and also the MPRO from SARS-CoV-2 for the development of treatments for COVID-19. So in our group, we have been working with several proteases, such as cruzaine, a target for Chagas disease, rhodesiine, which is the homolog, an ENT brucei, and a target for human African trypanosomiasis, and also viral proteases, such as NS2B, NS3 from Zika virus, and MPRO and PLPRO from SARS-CoV-2. In the interest of time, I'm going to focus on cruzaine, which is one of the proteases that we have done most work on. And it's a target for Chagas disease. Yesterday, you already heard about Chagas, especially in Santuza's talk. So I'm just going to briefly highlight that this is a disease that has been endemic in Latin America for over a century. And these days, it's also present in several other countries. And despite knowing the disease for more than 100 years, we still don't have any good drug to treat Chagas disease. There have been a couple of drugs approved, benznidazole and nifurtimox, but they are effective mostly on the acute phase, so we need better drugs that are effective at the chronic stage and also that have less adverse effects. And even when we look at initiatives that target developing drugs for neglected disease, such as if we analyze the portfolio from the DNDi initiative, we'll see that for some diseases, such as sleeping sickness, there have been a few successes in terms of implementing new drugs. For leishmaniasis, there are quite a few compounds currently at the clinical stage, but for Chagas disease, this pipeline has still been quite slow. And so far, the NDI has only reached the approval of a pediatric dose for benznidazole, and we don't have many compounds in this pipeline. So looking for new potential drugs for Chagas disease, we have been targeting cruzane. Cruzane has been a target for a few decades in terms of Chagas disease discovery. This is the major cysteine protease of the cruzi, and it's part of a multigenic family of cruzipanes. This protease is really important for the parasite for several reasons. It's important for nutrition, for invading host cells, for metacyclogenesis, and also to evade the macrophage response. And there have been several classes of cruzane inhibitors, and they have shown activity in vitro models and also in animal models of the disease. However, we still don't have any drug in the market that acts through this mechanism. Also, it's very, very important to highlight here that we have dozens of crystallographic structures of cruzane in complex with inhibitors, and that's important in terms of the research I'm going to tell you about, because we work with structure-based drug design, and we can use this information to rationally find new molecules that inhibit cruzane. Once we decide to work with target-based drug discovery, our next step is to find new molecules, and this is a big challenge, to find new ligands that target a protein. And when we think about new molecules, one of the first questions is what's the chemical space that is available, and this chemical space is huge. There is an estimate of at least 10 to the 60 drug-like molecules. This is a huge number. Really, the vast majority of these molecules have never been synthesized, but we know that there is, in theory, this space available that could be explored. So the next question is, in this vast chemical space, what can we do, right? Which molecules can we access, and how can we explore this space to find ligands? One of the alternatives is to do experimental screening. The idea here is that you can assemble a library and basically evaluate all of the compounds against your target. This is attractive in a way because you think that if you have a big library and you screen the compounds, you're not going to miss any hits, and you should be able to find new molecules, right? But it also has several problems, because when we think about these HTS libraries, you're going to find that maximum a few million compounds, and that can seem to be a lot, but if we compare to that number I just gave you, it's really a tiny fraction of the chemical space. And in addition to that, it's really labor intensive to work with the hits we find in experimental screening, and also frequently there is a very low hit rate. You screen all of these molecules, but you don't have the right molecules in your library. So many times you don't find anything interesting. And one alternative to that is to do virtual screening. Now the idea is that you're going to use a computer-based strategy to evaluate your whole library, and then you're only going to take to experimental screening a few prioritized compounds. This is very interesting in academia. Frequently, we don't even have access to these big libraries for experimental screening. And it's also interesting if you think about pharmaceutical companies in terms of saving a lot of resources and time to describe new inhibitors. We can do virtual screening based on the structure of ligands that are already known for a target. So I can look for molecules similar to the ones that I already know that work against my target, or they can be structure-based. That's really the focus of my group. And a big advantage of doing structure-based drug design is that I'm gonna look for molecules based on the structure of the target and find molecules that have good chemical complementarity. And that allows me to find compounds that are much more novel and that differ from the ones that are already known. So today I want to give you a few examples about the strategies we have been taking depending on the size of the library we have and the resources we have. And in one of our projects, we decided to do experimental screening. This was possible because it was a small set of compounds, 400 compounds, and it was a especially interesting set of compounds provided by the Medicine of Malaria Venture. So we tested all of these compounds in the assay against cruzaine, did an extensive work of validation to eliminate the false positives, and we got 4 confirmed inhibitors here. I'm highlighting one of them, which is shown here. And we saw that it had a competitive mechanism of inhibition, which is coherent with binding to the active site. And we proposed the binding mode for this compound using docking. We also shown that this compound was selective for cruzaine when compared to mammalian catepsins, and that's important in terms of safety, and also that this compound was quite novel. What I'm showing here is basically a heat map for chemical similarity. We are comparing the compounds pairwise, and the closer this is to blue, it means that compounds differ from each other. So we see that the new molecules that we found that are shown here are very different from the other known cruzaine inhibitors. And having this compound in hand, now we wanted to optimize this molecule. And to do that, we collaborated with Professor Saulo Andrade's group at WURGS. And Elani and Deborah were the students working in this project, in which we aimed to plan new analogs for this molecule, synthesize them, and evaluate them. And here, we wanted to explore the chemical space around this molecule and wanted to use these computational techniques to help us in this process. So the first thing we did was to think of how this molecule could be synthesized, and we basically breaking it here into the components that would be used, and that allows us to think of reagents that are commercially available and to assemble a virtual set of analogs for our hit. This this is highly effective in terms of generating analogs. So based on 12 central cores and 74 amines, we generated a set of almost 300,000 compounds, which we virtually screened. And then we selected 22 compounds for synthesis and characterization of their biological activity. This kind of project allows us to get SAR, or structure-activity relationships for these compounds, which basically means we can understand which parts of the molecule are essential for activity and where we can make modifications to try to improve properties. And I think the details are not that important here, but I want to highlight we identified a central core of the molecule, and in these rings, we are also able to identify types of modifications that were viable and led to the discovery of this new compound. Similarly to the original hit, it was also a competitive inhibitor of cruzaine. We, again, proposed its binding mode based on docking studies, and this compound was quite interesting because it eliminated a undesirable property that we had in the original hit. Also, when we evaluated against T-cruzi, the compound was quite potent, and if I don't think you can see it that well here, but when we compare to the control, we basically don't see amassed goats in the treated samples. And the compound was 15-fold more potent than benzinidazole in the same assay, and also it didn't show any toxicity against myoblasts. So I told you about a strategy to find a compound and optimize it with virtual screening, but we also wanted approaches in which virtual screening was applied to find new classes of molecules. Here, the idea is that when we do virtual screening for ligand discovery, for each compound in the database, we are going to explore several possible binding modes for each of the molecules doing the docking. And then we are going to rank our whole database based on the predicted docking scores. And the program should be good enough to enrich the actual ligands among the top scoring molecules. We look along this set to select molecules for testing. And this was the project led by Viviane during her PhD. We started with a set of almost 400,000 compounds, which were all commercially available. This is important because afterwards we need to test the compounds that we find most interesting. And we applied a pipeline with a couple stages of docking, and that leaves us with that list of compounds classified based on their score. But now we have to select them through a visual inspection of the binding modes, and we wanted to explore all of that information I told you that is available about cruzaine. We have several complexes of cruzaine with inhibitors, and we can learn from this data by seeing how these molecules usually interact with the target. To do that in a more automized way, we used Napoli, which was developed by Alexandre, a PhD student in the bioinformatics program. And the idea here is that based on a reference PDB, we search for all of the cruzaine complexes, and we get information about what's the frequency of interaction with different residues, and we use this filter to identify, to establish criteria for selecting compounds. And after finding a set of 17 compounds, we tested them, and identified compound 8 as a new competitive inhibitor. We also did molecular dynamic simulations to better understand the binding mode of this compound, and again, design analogs, and found a new analog that was not, it didn't have such a high inhibition of cruising, but when we tested the compounds in vitro in collaboration with Professor Fabiana Machado's group, we saw that the new compound was actually more potent against amastigotes and triple mastigotes than the original hit. Okay? We have all of these approaches that led to new ligands, but so far I told you about still a limited set of compounds that were evaluated even by virtual screening, and what we have been seeing lately is that the available space of commercial compounds has been really increasing very fast, and we are interested in this larger chemical space because it has been shown that it allows to identify higher hit rates. This is a comparison done by the same group against the same target, and when we see that when they worked with a bigger library, they found a hit rate that was 3 times higher, and also this approach leads to compounds with better docking scores and higher potency. Also, they lead to molecules that are less similar to the available biological space, indicating that there is more novelty. Sorry about going a bit fast, but I'm close to running out of time. But the idea is that we know that these big libraries have a large potential, and we want to explore it. The question is how? Because even having docking as a quite fast computational approach, when we start thinking about these huge libraries with more than 1 billion compounds, it's too slow. There have been some studies in the literature in which the groups do brute force docking, which is really docking all of these molecules, but these type of studies are very computationally demanding, so they are not easily accessible, and then we need novel strategies to explore this space. One of them is to combine artificial intelligence with docking. The idea here is that when I have a compound library, instead of doing docking of all of them to rank the molecules, I can take a subset of this library to train a machine learning model that will predict the docking scores, and after that, I will only dock the compounds that are already predicted to have the highest scores. And that saves me a lot of time. And I still dock the most interesting molecules. So we have been employing this approach against cruising. This is the work of a postdoc, Stella. Basically, we found a data set of cruising inhibitors, generated negative molecules, decoys, to train our protocol, and we developed a protocol which was able to enrich our ligands when compared to the decoys. And now we have, we started with a library of 650 million compounds, used 1 million molecules to train the model, built this machine learning model based on deep neural networks. We've shown that this model predicts very well the docking scores, and we have applied the model to find new potential cruising inhibitors. This is ongoing work. I don't have the final results yet, But my idea here today was to show you a few different approaches that can be employed for ligand discovery and optimization, and how we selected different strategies based on the size of the libraries that we are interested in evaluating, and that they have been successful for the identification of novel hits. With that, I want to acknowledge all of the network of collaborators. I've highlighted the ones that were more involved in this project through the talk, and to thank all of my group. And again, thank you for your attention, and thank Ricardo for the invitation to be here and participate in this symposium.
Thank you, Rafinha. So we have time for questions. Maybe I can start. So you've done all these different strategies to find hits against the same basic protease.
Yes.
The first one you showed is very different from the previous available inhibitors, right?
Yes.
But what about the different searches you did? Do they come up with similar hits? So what's different between the different strategies? What do they bring? Do they come up with similar chemical composition, or they come up with completely different compounds that inhibit the target?
We look for completely different compounds. This, I went fast through I didn't give much details, but this is one of our criteria to prioritize the compound. We are not going to test the molecules that are similar to the already known inhibitors.
But do they inhibit, but do they target a different site, or they still inhibit the protease at the same site?
Hopefully at the same site. So for the ones I've shown you, we saw that they were competitive inhibitors, which indicated they bind to the same site. And I said hopefully to the same site because when I do the docking, I am focusing on the active site. So if I find the inhibitors to another site, it would be just serendipity.
Very nice, great work.
Thank you.
For the first inhibitor, that looks very promising for the in vitro. So do you have an idea, there are three questions, whether they target other members of the family of the cruzaine or the cruzipine? Second, do you have an idea if you can produce large amounts? And have you had the chance to test in vivo?
Okay, so starting by the last one, so far we haven't tested in vivo. Concerning making large amounts, I'm afraid I cannot answer that. And I know that the large-scale production can be quite tricky. I can say that from a small-scale synthesis perspective, one of the reasons we chose that class is because the synthesis was relatively easy. But I cannot speak about the scale-up. In terms of testing against the other member of the family, we are very interested in this because I didn't show the details today, but a few years ago we have described a few subtypes, new subtypes of cruzipines. We still don't have the enzyme, but we are collaborating with Professor Ana Paula from UFRJ, and hopefully soon we are going to have active enzyme and start testing these compounds against the panel of the different members. And this is very relevant also because cruzaine is expressed more in epimastigotes, the subtype 1, but we have shown, at least based on RNA sequencing data, that the other subtypes are more common in triple mastigotes and amastigotes.
Well, thank you very much, Rafaela. Okay, so now we have a 15-minute coffee break, and we'll be back here at 10:45 for the second part of the session. Thank you.